Hi, I'm Larry Johnson, and today we're going to talk about what makes up cells. We might have remembered before when we talked about the different components of the cell, you have a cell membrane, which is the gatekeeper. That's the gatekeeper, that's what surrounds the cell and maintains it from its environment. We also talked about the nucleus. Nucleus is kind of the computer. It regulates things, tells things based on the DNA of what a cell should do, what kind of cell it is. Mitochondria is the power plant. That's the one that makes ATP, which is the cellular energy of the cell. Then we have the endoplasmic reticulum, and that's the construction team. That's the one that makes things, makes proteins for secretions, or it may detoxify if in case of smooth endoplasmic reticulum. And we talked about the Golgi. The Golgi is a packaging department that we have. But today we want to do a little differently. We want to look at the cell. Here's one cell here, and there's the various organelles that we're going to talk about. A little more detail about the organelles, and we're going to relate it to what you see at the light microscopic level, here and here, and also the electron microscopic level. So we will take these structures out of this cell and show how they fit into light and electron microscopy. Now, what makes up a cell? The nucleus and cytoplasm. So we see this cell, here's this nucleus, and then this is its cytoplasm around it, and then the cell membrane surrounds that. What makes up cytoplasm? Cytoplasm, there's three major components that we'll talk about, as well as the cytosol. The cytosol is really the liquid matrix inside the cell membrane. So you're inside the cell membrane, but outside the organelles. So it's the liquidy protein substance that surrounds the organelles and the inclusions. But we have three major classes or groups of structures inside the cell. One is the membranous organelles. That is, they have membranes. If you got membranous organelles, you probably also have non-membranous organelles. They have no membranes, is what we have. And then the third component are the inclusions. These are things that are expendable. They need not be in the cell for that cell to live, but is important for that cell function. So we'll talk about each one of these, of the membranous organelles, non-membranous inclusions, and the cytosol, which surrounds these structures. So if we look at membranous organelles, like I say, they have membranes. What are they? Well, these are common structures with metabolic function. In other words, most all cells have to have them to be alive. Cell membrane is one of them. Cell membrane, remember the gatekeeper. And if we look at that, we can see this cell, and there's a cell membrane that surrounds the cell. We also have rough endoplasmic reticulum. Rough endoplasmic reticulum, remember that was a construction team, a series of flattened structures with ribosomes on the surface that makes it rough. If you got rough, you also must have smooth. And here's smooth endoplasmic reticulum, a series of branching and anastomoses to tubules that we have in the cell as well. There's also a Golgi. The Golgi apparatus looks like a series of pancakes with little vesicles coming on and off that we see. We have mitochondria. Mitochondria provides energy. That's the powerhouse, remember, and it provides energy, and we can see it has double membrane that we can see. And then there is a lysosome. This is a lysosome, has a membrane around it as well, and we'll see what they do. Because as we mentioned before, these have metabolic function. So they have a function. What is the function of these? Well, the cell membrane contains the cytoplasm and the nucleus, and it interacts with the environment. So it talks to the environment. It receives hormones, growth factors, maybe time of cell division, whatever. It receives messages from outside, and it contains the cell. We also have rough endoplasmic reticulum. The rough endoplasmic reticulum is the one that makes what it does. It produces membrane-bound proteins for secretions. That's what the rough endoplasmic reticulum does. We also have smooth endoplasmic reticulum, smooth ER, and that one detoxifies. You have that in your liver to detoxify it and also produce steroids like in the adrenal that you have. Another structure is the Golgi. Remember the Golgi, flattened vesicles with a lot of vesicles coming in and out? That's the packaging. It also adds sugars to proteins that are inside as well as proteins in the membrane. There's mitochondria. Remember the powerhouse? Mitochondria produce energy and it also produces body heat. Mitochondria can get up about 50 degrees Fahrenheit. Our body weight is 37, right? 50 is kind of hot that we can see. There's also lysosomes. Lysosomes are also membranous organelles, and they are internal digestion. So they digest organelles inside the cell so it can remodel, modify itself, renew itself. Likewise, it kills bacteria if it's in the white blood cells that we can see. And so these are the membranous organelles. As we know, it's a cell membrane. It contains a cytoplasm. The rough ER produce secretory granules. Smooth ER detoxifies. Golgi packages, adds sugar. Mitochondria provides energy. And renewal is a lysosome. These are the membranous organelles. There are also some that are non-membranous. They have no membranes. These are used as cytoskeletal components, the structural things that are responsible for the shape, as well as free ribosomes that we see. So microfilaments is one of those cytoskeletal components, and it's a rod. And here we can see the rod shape, and that surrounds the cell membrane. So it's all around the cell membrane. It projects up here to amplify the cell membrane. There's also microtubules. Microtubules are hollow tubes, like a straw, and you can see them radiating from this centriole. And if you had several microtubules, you see this one has 27 microtubules, around here 9 times 3 is 27, then that makes a centriole. And here we can see the centriole where other microtubules radiate from. Another rod is an intermediate filament, about a 10 nanometer filament, and that one is associated with structural components that's inside the cell that's associated with junctions, a certain junction. This is a desmosome or a hemodesmosome at the bottom. And then we got free ribosomes. They produce proteins within the cytosol. In other words, within that liquid inside there. The other component is the conclusions. The inclusions, expendable, need not be there, very important for the cell function. One is a nutrient. Glycogen is a nutrient. Lipid is a nutrient type of thing. Another one is pigment. And so the pigment, melanin. Here we can see melanin granules. This is the melanocytes produce melanin. It passes on to your skin cells to give you your coloration that you have. There's also secretory granules, zymogen granules that are being produced that we see in through there. Secretory granules, those are inclusions too. Very important for the function of the cell, but not important for the life of the cell itself. So if we look at some of these with electron microscope, we can see the intermediate filaments. They are associated with the desmosomes. You can only see a fuzzy thing up through there. We can see secretory granules for sure. Here we see secretory granules, lots of those. There's some that are less dense and some that are mighty dense. Then we see the Golgi apparatus for a series of vesicles that we in through there. Looks like a bunch of pancakes stacked up. There's mitochondria. The mitochondria we can see right here. Mitochondria produce energy. And then we have the lysosomes. Lysosomes, there's one there. There's another one right there. Then we have the cell membrane. Cell membrane limits the cell, as you can see. And there's rough endoplasmic reticulum. Lots of rough endoplasmic reticulum that we can see all through there, which is characteristic of a cell that produce a lot of protein for secretion. Rough endoplasmic reticulum that we can see. Now, if we look at the cells at the light microscopic level, we can see the secretory granules here. You can see these granules here. You can't see the membrane around it, but you can see the granules there. Those are secretory granules, which is the inclusion. We also see a lighter area there right around the nucleus, and that's the Golgi apparatus, usually a clear region that you can see at the light microscopic level. And then here we can see the blue tint. Can you see this is red and then this is blue at the base? This is blue because the ribosomes are so much rough in the plasma reticulum. The ribosomes on the rough in the plasma reticulum. Cell membrane, you can't see the cell membrane, but you can see the limit of it at the light microscopic level. If we look at cytoskeletal components, we can see the rods. These are rods here. These are microfilaments. These are microtubules that you see, the soda straws that are there. And there's intermediate filaments. These intermediate filaments associated with the desmosome. And here we can see the size of things. There's 5 to 9 nanometers, 25 nanometers, or 10 nanometers for the intermediate filaments that we can see. If we look at another light microscopic view, you see several cells. This is one cell here. That would be another cell there, another cell there. And if you look at that, you see that this cell, intestinal cell, has amplified surface area. That's what the fuzzy coat that we see. And the actin filaments are pushing there that makes that structure there. So we can't see the actin filaments. We can see the result of the actin filaments. And then this is the cytoplasm and the nucleus. And the nucleus has a nucleolus that we can see there as well. And then in these, we can see mitochondria. These structures here, these heavy blue structures, are mitochondria that provide energy. We also see secretory granules in the goblet cells. So this is the intestinal absorptive cell. This is the goblet cell that produces mucus that we can see. Now, both of these cells get together, and you can see multiple cells here are joined by junctions. So they have a occluding junction that's located right in through there, and there you can see where this cell attaches to that cell. And that's the characteristics of epithelium, is that they hold hands, and the hold hands that allows them to produce a cavity that we can see. Now, if we look at the nucleus, these are nucleus of different cells. So this is a funny shaped nucleus there. There's another one, nucleolus. This is another round spherical nucleus with a nucleolus. A nerve cell here has a big nucleus with a nucleolus. Here's a pancreatic cell that produces zymogen granules. It had a lot of rough VR we saw. It has a big nucleolus for producing ribosomes, because the nucleolus is what produces the ribosomes. And this is in an egg. So this is the cytoplasm, and then this is the nucleus and the nucleolus. All these. This is in a pituitary cell. These are granules that secrete. This is the nucleus and the nucleolus. So that we can have that. Metabolic activity is indicative of the size of the nucleus, the amount of euchromatin or lighter staining, and the size of the nucleolus that we can see. Now, if we look at the Golgi apparatus, we can see a series of pancakes that we can see with vesicles moving in and out. And this is the inverter phase, where you have vesicles moving in through here, and they move through as they modify and expose the enzymes to add sugars. And then we have the trans phase, which is there. So this is the cis phase and the trans phase of the Golgi apparatus that we can see. There's also the nuclear envelope. So this is part of the nucleus. You can see the heterochromatin, euchromatin, and this is the nuclear envelope that we see there. And we can see nuclear pores. There's nuclear pores here. Also, we see ribosomes. These are ribosomes in through there, about 20 to 30 nanometers. And we can see glycogen. Glycogen, about 90 nanometers, and they usually form rosettes or clusters. Then here we see Golgi again. There's one Golgi apparatus, another Golgi cis and trans phase. There's a nucleus in through there. There's a nuclear pore. These are lysosomes and mitochondria. And there's a rough endoplasmic reticulum, and there's free ribosomes that we can see as well. Free ribosomes, and the rough VR is a series of vesicles with ribosomes. In this slide, we see some various cytoskeletal components. First of all, we see the microfilaments, rods, remember, and this is the cell surface in through there. And so we have a lot of microfilaments just below the surface of the membrane that we can see. We also have some free ribosomes that are there. And remember, proteins produce, they produce proteins in the cytosol itself. And then we have mitochondria. There's a mitochondria right up through there. There's a rough endoplasmic reticulum, a little piece of rough ER. You can see the membrane there and the ribosomes on the surface. These are microtubules with a hollow straws that are projecting in through here. And then we have intermediate filaments. This is an intermediate filament here and here. And then we have, this is a nucleus, it's part of a nucleus here. And we have heterochromatin, which is wound up, inactive. And then we have euchromatin, which is active. And we can see that the microfilaments are indeed associated with the cell membrane, and that they follow underneath the membrane of the cell membrane. And so those give support to the cell membrane. And then we can see the free ribosomes. If we look at the microtubules are radiating from the centriole, and the centriole itself is composed of microtubules as well. And then we have the intermediate filaments, which are associated with desmosomal connections that hold the cells together. So this is the end of what makes up a cell. And we see that the membrane is the gatekeeper, the computer is the nucleus, mitochondria the power source, construction team, ER, and then the apparatus, Golgi apparatus is the packaging compartment. And we saw the various organelles and how they looked in both light microscopy as well as electron microscopy. Thank you.